Thank you. Welcome, everyone. Glad to be here today. I'm going to share my screen now. Okay. Well, my name is Dr. Matt Casson, and this is my first Balticon and my first sci-fi convention. I'm really excited to be here today to talk about an unbelievable science story without any fiction involved. I'm an associate professor of mycology and plant pathology at West Virginia University, and I've been there since about 2014. Before I get into the actual story about the cicadas, I'd first like to start with a question. What are fungi? And I know a lot of you that are watching this from wherever you are probably eat mushrooms, use them in your salads, maybe you grill up some portobello mushrooms. I think a lot of people, when they think fungi, they think mushrooms. Now, it's true that all mushrooms are fungi, but not all fungi are mushrooms. In fact, I'm sure a number of you can name a lot of different fungi. For example, you think of brewer's yeast, which is really commonly used for making beer and breads. Of course, a lot of you probably have yeast and work with that when you're baking. Blue cheese is a cheese product that utilizes a fungus to provide a very unique flavor, so you might be familiar with that. But the point is here that fungi are not just mushrooms, they're quite diverse. So what are fungi? So this is a large group of mostly filamentous organisms that form their own kingdom. These are separate from plants, animals, bacteria, and others. Despite what you might see in the grocery store, they are not closely related to plants. They're incapable of producing their own food, so they get their food from other things. They kind of act as these enzymatic drills that mine down into different substrates, whether it be bread or wood or even human skin. And they use absorptive nutrition, as we could see here in this little drawing here, where they secrete digestive enzymes. They break down the substrate, and then they basically absorb the more simplified sugars and things that are constituents of those larger compounds. Fungal cell walls contain chitin, which is similar to exoskeletons of arthropods, which I'm sure many of you are familiar with, at least insects. But kingdom fungi is hyper-diverse, and I think, I want you to go away with that as much as I want you to go away with knowing how fungi modulate cicada behavior. I'm sure many of you have heard about the chytrids that are killing off amphibians worldwide, the massive number of frog extinctions that have happened globally due to a fungal invader. I'm sure many of you have taken walks in the woods and seen conchs or fungi fruiting out of the base of a tree and wondered, oh, I wonder why they're there. Well, they're there because they are one of the few things that can break down that substrate. And of course, we've all left oranges and bread loaves and a number of things in our refrigerators only to be discovered with fungi growing atop them, whether it be the green penicillium-like fungus or the black bread mold Rhizopus stolonifera, as seen here in the lower right-hand corner. What surprises most people is when you tell them that fungi are most closely related to animals. Certainly, they share some common things. I mentioned that their cell walls contain chitin, and we know, for example, shrimp and insects, they have a chitinous exoskeleton. And again, part of this confusion about them being separate from plants but closely related to animals kind of springs from the grocery store shelf, where we often find them juxtaposed next to string beans and things like that. And we put them atop our salad, so we just assume they're plants, but they are far from it. They form their own kingdom. Now, this is a little grotesque, but being at a sci-fi convention, I figured everyone could handle this. Knowing that fungi, include many opportunistic parasites and commensals on humans. Many of us daily use a dandruff shampoo to battle a fungus that loves the oil glands of our scalp, and that's Malassezia, the dandruff shampoo. Unfortunately, some of us suffer from toenail fungus, which again is another fungus. many of us, whether it be our kids or at some point in our lives, have had yeast infections, thrush and a number of different types of yeast growing on our bodies in places they shouldn't be, or in abundance levels they shouldn't be at, because maybe we're taking an antibiotic that's killing off the bacteria. And of course, ringworm, which, you know, sounds like it's a worm, but really is a fungus. These are all opportunistic fungi, and kind of speak to the diversity of fungi that inhabit us. But, you know, one of the things that it's important to mention is that fungi are really hard to control on our bodies. If you think about dandruff, if you've been using dandruff shampoo for 10 years, you probably still have it. Why is that? If you have a toenail fungus, it's really hard. Because the fungi are so closely related to animals, it makes defeating them when they invade our cells really challenging. Fungi also have some really unique characteristics. I can go on for a whole talk about this, and actually part of this talk centers around some of these unique compounds that fungi are capable of producing. Many of you may know that fungi produce antibiotics. Penicillin, for example, was the first antibacterial that was mass produced around World War II. And this is a really important antibiotic to this day that kills off bacteria. And it's produced by this green mold that you often find growing on your bread if you leave your fresh rolls out too long. They also can produce some really unique compounds like ergot alkaloids. Maybe you've never heard of those, but I'm sure you've heard of some of the effects of ergot alkaloids. Ergot alkaloids cause something called ergotism, or St. Anthony's fire. This is a real problem during the Middle Ages. But more recently, it's believed to be intertwined with the Salem witch trials, as you can see here in the upper right-hand corner. Ergot is a fungus that invades cereal grains, and it replaces the wheat heads with sclerotia. And when we eat these sclerotia, these contain natural lysergic acids, like LSD. So it's believed that the Salem witch trials, for example, was just a case of a number of wet years that led to a mass outbreak of ergot on rye, a mass consumption of that ergot, and mass hysteria as a result of everyone tripping on essentially LSD. There's other compounds like Coprine, which can be found in this white shaggy mushroom you see in the upper left. This is a really interesting compound too, because it actually prevents the breakdown of alcohol to acetic acid. So if you consume these mushrooms in the presence of alcohol, even one sip of alcohol, you'll get alcohol poisoning. And in fact, it's so good at doing that, pharmaceutical companies have found a way to formulate this into a pill given to alcoholics that want to stay on the wagon. It's called Antabuse. And just one sip of alcohol on this medication results in alcohol poisoning. Of course, many of you may be on a statin or know of someone on a statin, have a parent on a statin. Statins were first, these are fungal derived compounds, first discovered in the 60s, 50s, 60s. And of course, they could do amazing things like degrade lignin, which is one of the constituents of wood. You think about the trees that grow in our landscapes and in our forests. When they fall to the ground, what's going to break them down into soil? Well, it's fungi that serve that role. Given how diverse fungi are, it probably shouldn't come as a surprise that certain fungi evolved to kill insects. Now, these are some really pretty examples that I wanted to show you here. We could see Ophiocordyceps on a cicada killer wasp on the upper left. We could see an Ophiocordyceps on a cicada nymph on the upper right. And then we have a fungus called Furia down bottom on a snipe fly on the bottom side of a leaf. Now, some of these fungi are specialists, meaning they only have one host, like this snipe fly pathogen. Some are more generalist, where they can invade caterpillars and flies and bees. So they're very broad in who they can infect. And then some fungi have a specific life stage that they infect. This gets me into the topic today, which is on cicadas and the cicada fungus Massospora. In 2016, a periodical cicada emergence happened in West Virginia. In fact, you can see a map here of all the, in the lower left-hand corner of all the places where the cicada emerged during the 2016 season. And basically you can see all the cicadas here in the right-hand picture. What I want you to notice is that all of them look normal, except for this one here. If you have your screen and you're seeing me, you might not see it, but one of these cicadas, you can notice that its backside or its posterior end looks like a chalky white rounded mass. And basically what you're seeing there is the fungus Massospora. And about this time that they started to emerge, two of my grad students at West Virginia University approached me and said, hey, could we study that fungus? And I said, I don't know the fungus. Even though I'm a classically trained mycologist and plant pathologist, it's not one I had encountered. And they said, okay, well, we'll bring you a sample. And they did. And this is one of the ones they brought me. And I said, oh, this is so fascinating. But I had no idea what the story was going to turn into. At this point, it was just find some cicadas, some of which had fungal infections, collect them, and maybe move forward and see what we find. To give you a little background, I wanted to talk about cicadas first. This is a superfamily in the insect order Hemiptera. Even though they look scary, you can see them on my student's hand here, they have sucky mouth parts where they feed on sap. So they're not going to hurt you. They're big and they might fly and get caught in your hair, and that might be scary, but they can't hurt you. There's about 3,000 species worldwide. A majority of them are in the tropics. Now there are annual and there are periodical cicadas. The story I'm telling today involves both, but the one that you may know here in Maryland is the periodical cicada that emerges. In fact, it'll be emerging next year, and I'll show you a map here in a second. But just because something's called an annual cicada doesn't mean that it takes only 1 year to go from being basically an egg to an adult. It takes several years to develop underground, but some part of the population emerge each year, therefore giving the name annual. Whereas periodical cicadas are synchronized, and they'll emerge in a given 2-week period every 13 or 17 years. And there's 15 extant broods, and I'll talk what a brood is. But the periodical cicadas emerge in the billions, into tens of billions. There's just a massive amount of them. So what is a brood? A brood is a population of 13 or 17-year cicadas that emerge in a given year in a specific, in a given area in a specific year. For example, there's 12 17-year broods, and there's 3 13-year broods. Now, if you look at the map here, you can see the different populations and where they occur and what brood they are. Just have you pay attention to the yellow there in Maryland, and that's brood 10, and that's coming out in 2021. So what I also want to point out is that some of these broods overlap. For example, I'm in West Virginia, which has many different broods. So we're lucky that we get to see them. You know, we don't have to wait 17 years in between seeing them because, you know, one emerged in 2016, one emerged in 2019. So we were able to see them just within a few years of each other. And the other thing I'll point out is that you could see that there's like a small population in upstate New York, up in the Syracuse area. That population is very small, and it's likely to go extinct over the next few 17-year cycles just because it's reached such a genetic bottleneck. So what cicadas are present in Maryland? Just because, you know, here we are at Balticon, I figured I'd bring this in, make it relevant. We have a number of cicadas. The neotibicin and megatibicin are something you probably know. They're these green ones. We call them dog day cicadas. We often hear them in the dog days of summer, so like the late part of the summer, and they just scream from the trees. So they don't occur in massive numbers like you see with the periodical cicadas, but they certainly are loud. They make up for it with volume. And then the Massospora, which are these periodical cicadas, the brood 10, which emerged in Maryland last in 2004, is set to emerge here in 2021. So, and this includes a number of species. But as I'll tell the story about Massospora, they are all susceptible to Massospora. So here's a family tree of cicadas, the same way you would think about your own family tree. And these kind of are done based on the genetic relationships among these cicadas. There's 3 genera that I'm going to focus on today, and I want to kind of show their position on the cicada tree of life. And those are indicated by a red arrow at the base of the node. So that's the periodical cicadas, which is the ones you may be familiar with, the wingbanger cicada, which is out west, and Say's cicada, which again is native here in the east, but less common, so you might not actually be familiar with it. So some more information about cicadas. They have piercing mouthparts for drinking sap. People say, well, how do they know when to come up after 17 or 13 years? Well, it turns out while they're tapped into a tree, they're actually counting sap cycles. So because we have, periods of time where trees are dormant in the winter, late fall through early spring, as the sap starts to run again, they are able to count up to 17. And that's really fascinating. There's a lot of studies into the mathematics of cicada emergence and prime numbers. And that's a topic for a different person at a different time, but it's certainly, if you're into math, there's a lot of papers on this. They have these obligate bacterial partners that live in their guts. They basically have a specialized suitcase in their abdomen that holds these bacteria. In exchange for a house in the abdomen of the cicada, these symbiotic bacteria help to synthesize essential amino acids that the cicada can't get from the sap alone. Sap is not very nutritious, but in combination with the bacteria that help synthesize these amino acids, it's plenty enough for them to thrive. And they have conspicuous courtship calls, which I'm sure many of you are familiar with, just because you've heard them loudly blaring from your tree. And if you look in the lower right-hand corner here, you can see basically something called a timbal. That is the vocalization organ that's only found on males. And basically the male abdomen, which you can see everything behind the male cicada, acts as a resonating chamber. So they're able to do this. And if females are receptive to mating, and this is an important point for the rest of the story, is that if females like the male's call, she'll flick her wings and signal to the male, you know what, okay, you can attempt to mate with me, I'm ready for that. Now, cicadas are susceptible to a number of insect-destroying fungi. I'm sure that many of you have lifted rocks or lifted logs and found insects in various states of decay, like you're seeing here. This fungus, Metarhizium, on the left is a generalist fungus that will infect and kill a lot of different insects. The one on the right, this Ophiocordyceps, is a really unique one. Very analogous to the zombie ant fungus that just erupts out of the head of the cicada nymph. You can see that the fungus itself is much bigger and longer than the cicada nymph itself. So we're currently working to describe this. This is a new species to science that we found out in Arizona. But if you were to cut one of these cicada nymphs open in half, like you can see here, you can see that it's completely filled with fungal tissue. So it's now just acting as a shell where the fungus can fully colonize as it depletes everything that's inside it. Excuse me. So Massospora is really interesting. This is formerly what's referred to as a Zygomycete. This is a group of sugar-loving fungi you often find in breads and things like that. But this group, subgroup of Zygomycetes, which is now in the Zopegomycota, as you can see kind of highlighted here in the red box. This is a member of obligate parasites that really attack arthropods. So really they can't be cultured, they can't be grown on a petri plate, they only really grow on their insect hosts. And a close relative that maybe some of you are familiar with is this entomophaga in the lower right-hand corner. This causes a summit disease in flies where flies that are infected with this fungus, they will fly to the highest point in a building, they'll position their abdomen outward on the glass, and then they'll explode. The fungus will explode from their recently killed cadaver. So very sci-fi like, except this is certainly more science than fiction. So with Massospora, there's at least 13 species of these sexually transmissible pathogens. Yes, these are sexually transmitted diseases in the cicada world. They were first described back in the late 1800s from a cicada in New York. But really, after they were first described back in the 1800s, they remained kind of an oddity, a mycological oddity for 100 years. No one studied them. And part of that is the thing I was explaining in the last slide, where they can't really be grown on petri dishes, so it's really hard to study them because of that fact. So for a long time, we relied on spore morphology and cicada host to kind of say, okay, this is this species of Massospora and this is the other species of Massospora. But you know, it turns out that spore morphology oftentimes will overlap. So it's really hard to say, okay, this is one species based on a spore being this big, and this is another species based on a spore being a different size. But with the advent of DNA sequencing that came about starting in the 1990s, we were able to understand whether or not those relationships that were resolved based on morphology actually held up. And in our recent study, we've actually found out that these two that are in this red box are actually the same species based on DNA sequences, even though they infect two totally different cicada hosts. So that was really interesting to find, and it actually plays into this study I'm going to be presenting here about the psychoactive compounds. So we know that Massospora life cycle is, well, we know that we don't know a lot actually, because the cicadas and the fungus spend most of their lives underground where we can't really observe them. This is kind of a detailed cartoon that we put together for a paper we just published in a scientific journal. And what you can see is that these nymphs, if you look at this box, you can see some cicada nymphs underground. They get covered with spores. They emerge here on the left side if we follow the arrow up. And then eventually their abdomen starts to slough off. These plates that make up the exoskeleton start to come off in rings, revealing this white fungal plug that's hidden beneath. So their abdomen is basically replaced by a mass of spores and fungal tissue that mimics the back end of a cicada, but is all fungus. And eventually those fungi make their way back into the soil to infect the next generation. But I think if you're going to leave this slide with one idea that's fascinating to me to this day, is that the fungus can persist underground for 17 years while it waits for its host. That's really quite amazing that a fungus can persist as a spore underground for that long and wait for its host to pass by to infect it. So I wanted to talk a little bit about behavioral modification, because after all, that's kind of what this story is about. There's something called active host transmission in these cicadas that are infected by Massospora. What does that mean? Well, instead of slowing down the insect and making it kind of groggy, you'd think if you had a raging infection or a sword wound to your stomach, you'd probably limp around and eventually just sit down and succumb to your wounds, right? Well, with cicadas and Massospora, we see that not only does the fungus maintain activity, it seems to almost accelerate it. There's kind of a prolonged wakefulness in these cicadas, despite these conspicuous infections and despite the fact that almost a third of their body has completely fallen off, including their genitalia. And this active host transmission is actually seen not only in Massospora, but in two closely related fungi called Strongwellsia and Entomoptera. But what's really unique about Massospora infections is this hypersexual behavior we see in infected cicadas. So males will continue to attempt to mate with females, even though the back end is a fungal plug, they'll continue to spread this. And in addition to trying to mate with females, they'll mimic females to attract males. And this is a strategy actually by the fungus. We call this an extended phenotype. The fungus has taken the reins of control away from the cicada and now has the reins in its hands and is basically driving the cicadas to come in contact with as many healthy individuals as possible. In fact, doubling the number of healthy individuals it comes in contact. And this is really present only in cicadas with these fungal infections. So really interesting. Now this is different than a host behavioral modification called active host transmission. This is one that if you've ever read about the zombie ants, that's what happens in this system. And the same with the Entomoptera. You can see that fly there in the far left with its wings up and what looks like a cushiony abdomen. That's all fungal spores that are rupturing out. That's summit disease, where the fungus manipulates them to climb to a very high point, affix to that point, and then die. After they die, the fungus sporulates from their cadavers. And then spores are disseminated through gravity and through rain splash and through active shooting of spores. Active host transmission is different. It requires a living host, and the host behavior facilitates the pathogen transmission, thereby increasing the pathogen fitness at the expense of the host. We can kind of see here on the right, this is kind of a flow chart for these different behavioral modifications we see with fungi and insects, where some, for example, Massospora, you can see it leading to active host transmission, and some of these other pathogens are summit disease, and then some don't cause any manipulation at all. But if there's a common theme that's here, it's that a lot of these entomopathogens have found ways to modify their host to benefit the dispersal of the fungus. In fact, Massospora and Strongwellsia are really good examples of this, but there are other ones on flies and green apple bugs and thrips. And you can see in all these cases, in the fly in the far left-hand corner, you can see there's a hole there at the bottom of the abdomen through which spores shoot. So this fly has an active fungal infection that's carved a hole in its abdomen, but the fly is still flying around as if nothing's wrong. You can see this green apple bug in the middle here is looking pretty bad, but it's moving around even though its abdomen is ruptured open and there's fungal spores spreading around. And the same here with a fungus, Massospora infected Periodical Cicada on the right-hand corner. So we like to look at the genetics of these things and say, okay, well, let's try to reconstruct the evolution of these things the same way that you might overlay eye color onto your own family tree and try to trace back, where do these blue eyes come from? We're trying to figure out where active host transmission came from. And as it turns out, it's a lot harder than we think because there's a lot of fungi out there that have yet to be discovered. I said fungi are diverse early on in the talk. What I didn't say is that there's hundreds of thousands to millions of fungi that have yet to be discovered. So it's really hard to trace back various traits in an organism if you don't have the complete family tree, or you don't even have a partial family tree. So we're trying to do that, but it's proving to be difficult. So we're trying to do the math to try to figure out who's responsible for behavioral modification. We're trying to put the pieces together, but this is not a simple story. But some recent work on the zombie ant fungus, as can be seen here, have kind of really pointed us in the right direction. Their work suggested that fungal compounds, these small molecules, similar to pharmaceuticals we might take for a number of things, especially psychiatric disorders, might change the ant's behavior. So that was really interesting to ask. So we thought, okay, well, what if we did a specialized chemical analysis on these fungal plugs from cicadas? So we did just that. We took the fungal plugs from the backs of these infected cicadas. That's this white plug that can be seen here in the lower corner. And we also took the posterior ends of healthy cicadas. Why did we do this? It's really important in science to have controls. A control will tell you whether or not a healthy cicada has the same compounds as can be found in the fungus infected ones. If it's not found in the healthy ones, then that tells us either the fungus is producing it or it's produced as an interaction between the fungus and the cicada. So that's why it's important to have those posterior ends of healthy cicadas in the mix there. And then we did this chemical analysis and we showed that each sample produced these really unique signatures. And you can see that up here in the chart. You can kind of see a fingerprint for these two Massospora species, which are the first two, and then the third one's healthy. And you can see it looks less like the Massospora ones, indicating that these are obviously more similar in their chemical profiles or their chemical fingerprints than they are to healthy cicadas. In total, we found about 1,176 differentially detected small molecules or chemicals in these plugs, but really I'm going to focus on two of them, one from each of these cicadas. So I'm in my bed one night at 2 a.m. It's 2017, and my grad student's working late. And he calls me at 2 in the morning. And I pick up my phone and I say, his name is Greg. And I say, Greg, why you call me? It's 2 in the morning. He says, you're not going to believe what I found. And I said, but Greg, it's 2 in the morning. Like, my wife's pissed. Like, I hope it's important. And he said, those cicadas you sent? I said, yeah. He said, well, the ones from New Mexico that you collected, the annual cicadas, are loaded with psilocybin. I said, psilocybin? He goes, do you know what psilocybin is? I said, yeah, the magic mushroom compound. He said, yeah, it is. So we found psilocybin, which is a psychoactive tryptamine that's produced in about 200 different species of magic mushrooms, we found it as the most abundant alkaloid in the plugs of these cicadas, which is really fascinating. And if you look at this kind of figure over here, you can see that was only detected in the annual cicada plugs and wasn't detected in the periodical cicada plugs, nor was it detected in the negative control or the healthy cicada abdomen. And it was 17-fold higher compared to those other things. So I'm like, oh, wow, this is it. So, I mean, are you sure? And he said, no, well, actually, no, I'm not sure. Because really the instrument we used to detect it only was looking at its mass. Similar to, you know, let's take two participants on this call. You may have the same exact weight, but you're not the same person, right? So similarly, chemical compounds can have the same mass, but it doesn't mean that it's the same compound. There's a number of ways you have to test that. And the first way is to actually compare the cicada fungus with a chemical fingerprint of a standard, an analytical standard that's made by a chemical company that you can purchase and then run through an instrument and kind of compare them side by side. So again, coming back to this concept of chemical fingerprints, you're basically comparing the chemical fingerprints. So what I'm showing here in the top two charts is that the standard and the plugs from the cicadas matched exactly. In fact, there were 8 fragments that matched. You only need 2 or 3 fragments to say for sure you have the same thing, and we had 8 exact matches. So basically, that was that. An interesting thing that came about is when I had to order these standards, I went to go check out from the chemical company where I ordered them, and it said, please enter your DEA controlled substance permit number. I thought, oh God, I don't have one of those. And then I called my student who's been doing this kind of instrumentation for a decade. And he said, no, don't worry about it. They probably have DEA exempt standards. They sell them all the time because they realize that not a lot of people have these permits. So just order a DEA exempt one. And sure enough, we were able to find one and compare it. And like I said, the result was that what we found in the cicada fungal plugs was an exact match to psilocybin. But what about the periodical cicadas? I mean, those were the ones that were in our backyards. Those are the ones that a lot of you are familiar with. Those are the ones that are emerging in Maryland next year. I said, Greg, did you find psilocybin in those? He said, I'll look again, but I didn't see it. But remember, I told you there was 1,176 differentially expressed compounds that we found in the fungal plugs that weren't in the healthy cicadas. I said, well, what else is in that list? Let's dig through. When we started digging through and, you know, going through these compounds one by one, Googling them, trying to figure out what they are, looking in the periodic or the peer-reviewed literature to figure out what they do. And he kind of stopped and looked at me like with a stone cold face the one day. And I was like, what is it? He said, have you heard of cathinone? I said, cathinone? No, I'm not familiar with that. Is that like psilocybin? He said, cathinone's an amphetamine. And we quickly Googled it. And, you know, cathinone is a natural amphetamine found from this plant in the Horn of Africa that's used in the Horn of Africa and also used in the Middle East. It's chewed mostly by the men there as a stimulant, and this plant contains this amphetamine, and we were detecting in these cicada plugs. Now, as you can see on the figure on the right, you can see that we detected it pretty high amounts in the periodical cicada, but we did detect a little bit in the annual cicada, so that was really interesting, but none in the healthy cicada controls. So again, we're looking for candidate compounds that could explain the behavior. If it's in the healthy one, it's probably not going to be our good candidate compound. But the fact that we found it here and not in the healthy ones was a good indicator. So again, you know, I went to Greg, you know, okay, what do we do? And he said, try to get a DEA exempt standard and we'll compare the fingerprints. And we fragmented in this instrument, and sure enough, we got 3 fragments that matched. And we had none in the healthy cicadas. So we have a match. And now we know we have amphetamines in the periodical cicadas, and we have psychedelic tryptamines in the annual cicadas. And then I said, okay, well, what's next? And he said, well, since we have these standards, we can actually figure out how much is in each plug. We call this quantification. So we were able to determine the absolute amounts. When you think about dosage, you know, think about taking acetaminophen or taking a pill. You know, they always tell you what the dosage is or the dose. We were able to figure out the dosage in these plugs. And we showed that psilocybin was present in both our cicadas from New Mexico and our second species, which I told you earlier, we determined based on DNA as the same fungus. So that was really interesting. But we only found psilocybin was quantifiable in these sage cicadas and not in the wingbanger cicadas. And the reason for that is that as these compounds are produced, they will go through these changes where they might add a certain group. You can see that this group right here is not on the same compound and it's being dephosphorylated. So these compounds undergo kind of changes in chemistry, which make them different, but still active. And we found psilocin was quantifiable in 82% of the other plugs, but not in the healthy cicadas. So again, coming back to these are compounds that are unique to fungus-infected cicadas, and they're found in most of them. So at the same level too, and you can see this is a nanograms per fungal plug. Now we also looked at the absolute quantification of amphetamine in these periodical cicadas, and yes, we found it in several populations, but it wasn't present in but about 30% of the plugs. And moreover, it was only quantifiable in 24%. So, why is it so much lower than psilocybin? Well, if you understand the activity, the bioactivity of these compounds, you would know that psilocybin, maybe a trip from magic mushrooms, might last a lot longer than taking something like bath salts, which is a synthetic cathinone. If you're not a person who's taken bath salts, I have not, and I never plan to. Some of you may be prescribed drugs for attention deficit disorder or ADHD. These are synthetic cathinones as well, so they serve a similar way. So think about the half-life of an ADHD drug, same kind of thing. And actually think about the function of an ADHD drug, because it's going to come into play here and trying to think about how these compounds are interacting to affect the behavior of the cicadas. So a question I always get when I talk about quantification is, can I eat that? Is there enough of a dose in those cicadas that I could eat it and trip? Well, I often say one does not simply eat a Massospora infected cicada. And the reason for that is not because I'm against the use of those things, but I told you this was just 2 of 1,176 compounds in there. We haven't vetted the other ones, and we have no idea what their function is. All it takes is one toxic compound in those cicada plugs to really ruin your day. So as much as these have recreational drugs in them, they have a lot of other things that could really affect you. But how then do we understand the role that these compounds are playing in cicadas? Our next step would probably be to dose them, right? Take purified psilocybin or take purified amphetamine and dose the cicadas. And that would be the next step we would do, except psilocybin and cathinone are regulated by the DEA. In fact, when we first discovered these compounds in cicadas, I was really nervous because you need permits to work with controlled substances, but we didn't know we were going to find controlled substances. So I have a letter here on the right. I wrote the most awkward email of probably my career because I thought I was going to get a SWAT team coming through my lab office, tasing me and stealing my cicadas. I was worried that we wouldn't be able to publish this or talk about it in an open forum because basically we were dealing with controlled substances, but the DEA gave us a blessing. That's kind of what the letter says, that we weren't intending to do something malicious with it. It was just a kind of an inadvertent discovery. But it's something you don't think about until you have to think about. So that was really interesting. Fortunately for us, there's previous behavior modification of arthropods in the 60s and 70s, before the DEA started to really tightly regulate a lot of these compounds. A number of people dosed ants and spiders and blowflies, anything you can imagine, with LSD and mescaline and all kinds of things just to see the effect. And as it turns out in the literature, low doses of amphetamine, similar to what we found in the cicadas, caused increased aggression in ants, and it altered the feeding behavior in blowflies, which is kind of what we're seeing, a suppressed feeding or suppressed drinking just to mate. And the psilocybin ingestion had a less observable impact, and that's been shown in previous studies where they've given spiders human doses of psilocybin, and it messes up their webs a little bit, but not too much. So, but the point of this again is, we can lean on the previous literature, but really follow-up studies are going to require DA permits and strict oversight. So basically, we found these compounds present in multiple populations of infected cicadas, but not healthy ones. And the known neurogenic activities of these compounds explain some of the behavior prior to death. Focused effort, like you find with an ADHD drug, improved endurance or increased stamina, and appetite suppression. But the activity level effects of these compounds can't explain all the behaviors, and it's likely that some of the other compounds that the fungus is producing, or some of the insect hormones that are interacting with these compounds, are acting, synergistically to create the basically modified behavior we're seeing in front of us. But likely these compounds contribute in some way. We just don't know. Do they contribute to 80% of the behavior, 50%, 20%? We'd have to really dose them to see. Now, we have some genetic evidence that these compounds are being produced by the fungi, and we do this through genomics and things like that. But the long and short of it is that we don't have a lot of data and that we have to generate more. And some of the compounds are missing, which may indicate that this is a novel biosynthetic pathway. But we're continuing to sequence the genome. We're trying to culture it. We're making field observations and doing a number of things, including searching for additional Massospora species. Because, we're hooked now, and we really want to, we've published 3 papers on this, and we're looking to do more. But overall, the vast majority of arthropods, including cicadas, remain unstudied. There's numerous ways to investigate these, but really collection and storage is key. And there's some amazing interactions happening around us, like this cicada thing we worked on right from my campus. 300 yards outside my office. So there's amazing things happening all around us that could be turned into science fiction, but science can be stranger than fiction. And I think I've made a case for that. So for more info on this, you can check out, Ed Young did a piece in the Atlantic about it, this parasite drugs, it's host with psychedelic chemical and shrooms. And Joanna Klein did an article in the New York Times, drug castrated and eager to mate, the lives of fungus infected cicadas. We'll continue to publish on this. You can follow me on Twitter for more on this if you want to keep following the story, or other weird stories. You may have seen that we injected peeps with marshmallows back in 2019 and got some press for that too. So with that, I'm gonna stop sharing here and then take questions. Okay, so let me go to Q&A here. All right, so I'm gonna answer live here. Can broods crossbreed if their cycles sync up and they're geographically adjacent? Yes, they can. In fact, there's a good number of researchers that are devoted to this question and are tracking the expansion and contraction and the overlap of broods and how they interact. And this is how basically new broods can emerge. So that's a really good question. But just like new broods can form, also broods that remain in isolation can go extinct. I'm gonna answer this. Do the fungi infect the tree? No, the fungi are obligate on the insects. The cicadas do hurt the tree a little bit. They feed on they'll feed on the sap underground, but when they come out in the spring, they'll lay eggs, they'll oviposit eggs in the branch tips. In fact, I encourage you next year when the cicadas emerge to just look for dead branches all around, because the cicadas will lay millions of eggs in the branch tips and it'll cause them to die. The trees won't die, but the tips of those branches will die. There's a question, do you have a link in the study to the study on the small compound in the ant fungus? We mentioned that in our cicada paper that we published in Fungal Ecology, and that's open access, so it's not behind a paywall. If you go to the journal Fungal Ecology and you put my name in, you should be able to find our cicada paper there, so you can look there. Is there a way to make a healthy living space in the naturalized habitat? Certainly, one of the things I think about is these cicadas spend 17 years underground, right? Feeding on the roots of trees. If land developers come in and take out all the plants, those cicadas that were there are dead, right? So it's really important to maintain forested areas, even in suburban and urban environments. Otherwise, the cicadas won't persist. And you don't think about them because they're underground for 17 years at a time. And you may not think, oh, well, taking out this patch of forest is going to affect something that's been underground for 4 years or 7 years or 16 years. Imagine it making it to 16 years and then, you know, someone removes a whole patch of forest. Not to say that natural tree mortality doesn't happen in the forest and cicadas die, but when you remove a whole patch, you're removing a whole population. Is there evidence that the bugs actually hallucinate, experience a high? That's a really good question. There's some evidence that they experience altered time perception or altered time. The wingbanger cicadas in New Mexico, they tap their wings to mate. They don't actually have a vocalization. So what they do is they'll call like this. And the males tap way faster than the females. When males are infected, they'll tap at an intermediate pace. So it's clear that their perception of their temporal or their perception of time is skewed. Whether or not that's due to other compound or if it's due to the psilocybin, I don't know, but it's certainly an interesting question that we'd like to dig into. But again, it would require the DEA permits and things like that. So besides the ergot fungus you mentioned earlier, are they're fungi, bacteria, or parasites that modify human behavior to promote their own reproduction? Oh yeah, absolutely. And the toxoplasma in mice and cats is a really good one. There's a worm that will infect crickets that motivates them to jump into water and drown themselves so the worm can fulfill its life cycle. There's a number of fungal manipulators. Well, think about, you know, rabies is a really interesting virus, right? In fact, I'm sure many in this room could speak to it better. They say that like Dracula is based on the rabies virus. I don't know if that holds true, but this is a virus that makes you salivate and bite into things. So therefore, like every bite, you're spewing virus particles. And it makes you hydrophobic, so you don't drink. So you're basically like you're keeping a lot of the virus in your mouth. And so that's really interesting. But there's more. I just can't, you know, think of those off the top of my head. But the horsehair worm is a really good one. We're in a suburban area. Our whole neighborhood community hit by emerald ash borer this year. Still have ash trees showing up dead this spring. Wanted to know how to keep the area healthy. You know, EAB, emerald ash borer, is a real problem, and it's gonna, you know, it took out the ash. Fortunately, you know, we're not great about diversifying planting of trees. Cicadas can feed on a number of plant species, but I would say the key to resilient landscapes is to diversify what you plant. You know, don't plant the same thing as your neighbor just because it's aesthetically pleasing. You know, diversify to ensure kind of you have a policy in place for the next big pest or pathogen.
We have about 2 minutes left, sir.
Okay. Fascinating talk. Are the folks studying how fungal parasites evolved? Yes, absolutely. In fact, we're trying to understand how they may be co-opting gene networks that exist in the animals themselves and are utilizing them to get signals to their brains or to their muscles. There's a lot of great work being done on the zombie ant study at Penn State by Dave Hughes and another student named Teresa DeBecker, who's at the University of Central Florida. Do we know what mechanism caused infected male cicadas to utilize female reproductive behaviors? Well, we don't exactly know that. It could be hormone-based. Certainly these fungi castrate the males. They castrate the females too. They remove the genitalia. And there may be a hormonal cascade similar to what we might see in vertebrates. I'm not saying that they're exactly analogous, but it's something to think about, is hormonal cascades and how that impacts behavior. Do we know, okay, let's see. Thank you so much. You did a very good job speaking. Thank you, I appreciate that. Are there any cicada predators who are susceptible? Yes, not to the Massospora, but I showed you a wasp that's susceptible to an Ophiocordyceps. So even the predators that will kill cicadas are susceptible to their own fungal pathogens because fungi, at the end of the day, are the best. And that's why I study them. So I think we're done there, we're done there, we're done there. Okay, I think I'm about out of time. So thank you again for your attention, and I appreciate your interest, and hopefully I'll get a chance to interact with you some other time. Thanks.